This next set of presentations that you're going to see are basically going to be a summary of the earlier videos that you watched, put in maybe simpler terms or given you some diagrams that might be easier to understand what's going on in these processes. But this is going to be about the beneficial processes, not so much focused on food like you were seeing with the bread making, cheese making, and beer making, but more with sewage and water treatment, and then the nitrogen cycle and the importance of microorganisms in each one of these different processes. I'm going to start off with water safety and wastewater treatment. I have a nice little diagram here of what a wastewater treatment might look like, where all the stuff comes in from houses, wherever it goes into the first vat, if you want to call it, and it goes through all these various things until it's pretty much taken care of. And here's what an aerial view of a wastewater treatment might look like. But we have some concerns with water safety. One of them is pathogens. Pathogens can make people very sick. They can even kill them. Of course, there's economic considerations when you get people getting sick. They miss work. It costs the companies. It's going to cost the person possibly in medications that they have to buy, and certainly in the discomfort and misery they get from these particular organisms. Another safety concern, which kind of goes along with the pathogens, is the presence of something called coliforms. And these are organisms that grow around body temperature, and they're defined as organisms that can ferment lactose, which is a disaccharide. This particular organism here is E. coli, which we have in our intestinal tract, and it's able to ferment lactose. And this particular agar that we're using actually has an indicator when lactose is fermented, and it gives you this really neat looking green sheen. But I'll explain a little bit more about why coliforms can be a concern in a slide or two. And then we're also concerned about what the contamination sources are. Are they biological? Are they more of a man-made kind of thing from industry? Biological can mean a lot of times pathogens, Giardia, Salmonella typhi, those sorts of things. Or it can be heavy metals from various industries, mercury, cadmium, zinc, that sort of thing, that can be harmful to human health. Now, with drinking water standards, the intent of the process is, of course, to eliminate the pathogens, if not all microbes, from the water supplies. And there's various ways of doing that. But what we consider to be the greatest risk is fecal contamination, because in the intestinal tract of humans and animals, we find a lot of different bacteria, and some of them are pathogenic. The more, of course, with pathogens, the more there are, the worse it is. We're trying to avoid the fecal contamination. Nobody wants to think about consuming fecal material from other living organisms. Some examples of the organisms that we would find in feces include Cryptosporidium, Giardia, Salmonella, and cholera. You may have heard some of these, some of them you may not have. Cryptosporidium causes a diarrheal disease that can last for a couple of weeks. Giardia, not diarrheal disease, but different manifestation than Cryptosporidium. And then, of course, cholera is a diarrheal disease, but it's so severe that a person can come down with it and die within a day. So we definitely want to reduce the risk of or eliminate fecal contamination from water. So these coliforms I was talking about, we actually use them as what we call an indicator organism. I mean, in other words, if we can detect coliforms, it's an indication that there has been fecal contamination, and that means that we need to take a greater look at it to see if there are pathogens in it. But the coliforms, like I was saying, they ferment lactose at close to body temperature. Body temperature is 37 degrees. Coliform is defined at 35. They're going to produce gas as a result from it. Further description of it is they're aerobic, meaning that they're using oxygen. And when we get to looking at their cell wall, They're a particular kind that's called gram-negative, and we'll talk about that in another chapter when we get into bacteria. But just based on that definition, these organisms are not pathogenic. But if we can find them, it indicates there's been fecal contamination, which means that there is a higher probability of pathogens being in a particular sample that we're looking at. What I have shown here is the testing that we would do. We take the water that we're concerned with, we mix it in here and let it grow. This has lactose in it. If we have coliforms in there, they start producing the gas, and this little tube here captures some of the gas and is going to float to the top of the bottle, which indicates then that we have a coliform contamination. The little tube is referred to as a Durham tube, and this is lactose tryptone broth. Like I said, it's got lactose, but it's also got tryptone, which is a source of nitrogen for the organism. So the EPA has set a standard of 0 coliform bacteria in 100 milliliters of water. Normally when we put stuff out on agar plates, we would put 1,000th of that. We'd put a 1/10 of a milliliter instead. So in order to check it for coliform in 100 milliliters, we have to go to this larger system here to do that. Now, there are two different numbers that we can come up with if we do actually put them on plates. That's called total coliforms, and then there's the fecal coliforms. So the total coliforms is all coliforms. There are some that are fall into this category that we don't find in the intestinal tract and aren't eliminated from an animal in the feces. So that the fecal coliforms would be considered to be a subset of that. So if you want to look at it in some kind of a diagram, here we have the coliforms. Within that we have the fecal coliforms. Then we have a subset even of that where we have the coliform that's pathogenic. Now there is E. coli, or a lot of E. coli, that's not pathogenic, but we do have strains that do cause a lot of problems, and there's one that's called O157H7, which is a fecal coliform, but it's also a pathogen. So what this is saying is not all coliforms are pathogens, not all fecal coliforms are pathogens, but there are a subset of fecal coliforms that are pathogenic. And those are the ones that we're most concerned with. Now, some of the contamination sources, this is to represent a river or a stream. We can get fecal contamination from the wildlife. That's not a surprise. They're not worried about it. They don't, the fact that they're contaminating a water source is not part of their daily routine, worrying about that kind of thing. This is representing towns and cities. They will usually treat their water, but you can have situations where you get runoff. Let's say you have flooding. If the sewage treatment plant gets flooded, you get all that material, and it will eventually get into the river. And there could be other scenarios as well. You get agricultural runoff. Could be either fertilizers. It also could be the fecal wastes of animals, like on a dairy farm or a turkey farm, whatever you call them, that sort of thing. There could be contamination from it as well. And then we also have waste that's eliminated from our households, and if they're not treated properly, they can get into the water system as well. Or if you're living near a stream, you fertilize your yard, or you have animals, your own pets, fecal contamination could get into those water systems as well. Now, what's interesting is that we have the ability to determine the source of bacteria to some extent based on their profiles. And the profile specifically that we're looking at is antibiotic resistance. Because humans have been taking antibiotics for a number of years, we tend to have bacteria that are resistant to antibiotic, whereas wildlife usually doesn't get exposed to antibiotics. So if we find fecal contamination, we can take a look at it and then characterize it by whether it's resistant to antibiotics or not, and that can tell us something about where it comes from. Now, some farms do use antibiotics in the raising of their animals. Some of them use it to boost meat production, but some of them are only using it when the animal gets sick. But you're going to have different profiles from that, because if you're using it prophylactically or to boost production, you're going to be using a lot more than you would if you're just treating one or two animals. So we can use that to help us sort of pinpoint where contamination's originating.